Alright, so we are moving on to the third line of defense. So now this is when we get antigen specifics. So in the it's actually called the immune system now, and we're actually targeting specific viruses or bacteria, and so it's going to react and act against those. It's systematic, and so it's not just that initial infection site. It'll go all over the body. It has memory, and so that's the biggest difference between the specific versus non-specific. It has that memory. It recognizes, mounts a stronger attack on those pathogens. So there's two different types of immunity. The first is humoral, and if you remember talking about humoral before, it's just the blood, and so it's all our fluid. So this is antibody mediated. Cells produce chemicals for defense versus cellular, and so obviously that'd be cell mediated. So cells target specific virus infected cells. As a reminder, you need to remember what an antigen is. It means anything that's not itself. This could be pollen, it could be any of the pathogens, anything that is not yourself. And so it's going to provoke this immune response. Sorry about my moaning dog. So examples: proteins, nucleic acids, so different DNA, carbohydrates, all kinds of different things. There are some self-antigens, and so we have many surface proteins. If you remember in the video, it talked about that's how viruses actually get in, is we have our own proteins, and our bodies want to let our own proteins in, but the virus kind of tricks us. Our immune cells should not attack our own proteins, but our cells in another person's body could trigger you immune. And so this could be anything from cells to different organs, and so that's why you can't get transplants very easily. This is just showing a picture of how blood is made. And so remember, everything's made in the bone marrow, and you have these different stem cells, which just means it creates more like itself. So you have the myeloid, which is going to get your red blood cells, your platelets, and these other different types. And then you have your lymphoid, which is really where we're going to concentrate. So the lymphoblast, which makes your B, T, and natural killer cells. If you remember, the natural killer cell was part of the second line of defense. So the cells of the immune system, and remember, the immune system is just that third line of defense. It's not technically everything else. And this originates from the hemocytoblast, so hemo, blood, cyto, cell, and the red bone marrow. So they all actually come from red bone marrow. However, the B lymphocytes become immunocompetent in the bone marrow. That means that's when they're able to have their immune response, so that immunocompetent. What the B cells do is they produce antibodies. This is the humoral immunity. Then you have your T lymphocytes, or T cells, which again, they originate in the bone marrow, but they become immunocompetent. So this is where they actually become available for the immune system in the thymus. So notice B for bone marrow, T for thymus. And this is the cell-mediated immunity. Alright, so we have stuff attacking. So this is our macrophage. Again, big eater, arise from the monocytes, and they're all over the place. They like to go and attack everything. This is just going over again everything. So you have, they're all starting out here in the bone marrow, except for B cells. After going to bone marrow, they're still actually become the bone marrow, and then they'll go to the lymph node, whereas the T cells go to the thymus and then move over to lymph nodes and everything, and they'll just kind of recirculate there until something comes along and says, hey, you have an antigen, and then they'll make that final differentiation into actual mature B and T cells that can be used for the attack. So we're going to first go over this humoral antibody median, which means we're going to go over B cells. So again, B cells are the humoral. They're dealing with the blood. So their function is to manufacture antibodies, and they are awesome at it. And create a memory cell. This whole thing wouldn't work if we didn't have memory cells in case we got this same pathogen again. So how B cells are developed. This is getting really long. We've pretty much covered it. Don't get too bogged down with everything. So stage 1, you have immature B cells, again, that are within the liver and bone marrow, and they are going to synthesize or make one specific antibody. So if it's for Staphylococcus, it's going to make an antibody against just that Staphylococcus. You have to have another one for Streptococcus, another one for the influenza B virus, and all of the different flu ones. And so it inserts those particles into the cell membrane. They then migrate to the lymph nodes and wait there until that antigen, if you remember the dendritic cell, bringing it over until that antigen is presented. Stage 2, so that antigen is presented and it matches the antibody on the immature B cell, and it's now activated. And so these activated B cells start rapidly dividing, and most of them will become plasma cells. Those plasma cells are going to produce those antibodies to destroy antigens. And like I said, they are extreme factories. They can actually produce 2,000 antibodies per second. The activity lasts for 4 or 5 days. That's usually plenty of time to get everything. Some of those B cells are actually going to turn into memory cells. And so that, if you get Streptococcus strep throat again, it can have those memory cells to make that attack on it even quicker. So this is just showing in picture form what we just talked about. So here's your first encounter. It has whatever antigen from the pathogen that came in. It's going in, and it's making these immature B cells. Once it has that, they turn into plasma cells, which are making the antibodies against that, and some memory cells that can remember. So secondary, like it says, can be years later. We get those exact ones. We have the B cell that's waiting for them, and it's going to do the same thing. Plasma cells go, and we're going to make some more memory cells. As far as immunity goes, we have the naturally versus artificially, and active versus passive. So naturally, obviously it's going to be natural. So if I get sick with the flu, that's going to be natural. That's the active infection. I have contact with the pathogen. You can also get it naturally if you were breastfed from your mother. And so the mother goes through fetus or in milk. So those are the only way to naturally get it. Artificially, which to be in school, everybody has done this too. So you can get it artificially by a vaccine, dead or basically injured pathogens, or passively through a serum, which doesn't happen that much. And then you have different antibodies. We're actually not going to go into much detail, but I want you to know these antibodies are actually called immunoglobulins. There's all kinds of different ones, but you don't have to know that. You just need to know that they exist, and then you have and what they are. So they're proteins secreted by the B cells, which technically they're secreted by the plasma cells, and they're carried in the blood plasma, and they bind specifically to an antigen. All right, that gets you through pages 6 and 7, and so we are going to end the video now.